Welcome everyone. Today's talk, efficient generation of gene-edited mouse models and cell lines using synthetic single-guide RNA. My name is Gurpreet Balrey, Head of Business for Europe, Middle East and Africa for the Genome Engineering and Modulation franchise within Merck KGaA Darmstadt, Germany. My colleague, Dr. Peter Romanienko, the Managing Director for the Genome Editing Shared Resource at Rutgers Cancer Institute of New Jersey, is going to be joining me. He's going to be doing part of the presentation. Before we begin, just to let you know, the life science business of Merck KGaA Darmstadt, Germany operates as MilliporeSigma in the US and Canada, and Merck elsewhere, just so you are aware, because we are called different things in different places. In terms of agenda, I'll cover Merck's background in gene editing and introduce some basic CRISPR concepts, followed by an introduction to our sgRNA synthetic guide system. I'll then hand over to Peter for his section on their experiences of using our sgRNA synthetic guides in their animal model creation. Peter will then hand back to me, and I will conclude the talk with some new data we have on electroporation and microinjection of our sgRNA into mouse embryos, and a couple of data points on the use of our sgRNA in cell-based assays. We'll then have, hopefully, ample time for your questions. So in terms of background and introduction, Merck has been heavily invested in gene editing reagents for almost 15 years now. We began by supplying gene editing reagents for prokaryotic research back in 2005, as you can see on the left-hand side, with a system called Targetron. 2 years later, the next big development was supplying zinc finger nucleases, commonly referred to as ZFNs, where we commercialized products and services under an exclusive license from Sangamo as the first targeted gene editing reagent for mammalian use. Moving forward to 2012 and beyond, we're now firmly placed in the CRISPR era, and we continue our commitment to supplying cutting-edge gene editing reagents for model systems of any kind. Along the way, we've hit many firsts: the first genome editing tool in zinc finger nucleases, as mentioned, the first genome edited rats with zinc finger nucleases, the first to show utility of oligonucleotides as donors for knock-in, the first to have a commercial CRISPR product on the market in 2013, and the first to have a whole genome human and mouse lentiviral arrayed library. On the patent front, we now have 26 global foundational CRISPR-related patents approved. All in all, we have a depth of knowledge, expertise, and suite of cutting-edge products that is unsurpassed. Now, when we talk about gene editing in the present case, what we're really referring to is the targeted cutting of DNA at a specific predetermined site to increase mutagenesis frequencies at that particular point. Gene editing tools, whether they be ZFNs or CRISPR, are designed such that based on a target sequence, a nuclease can be brought to that position and cause a double-stranded break. Once this occurs, the scientist relies on the cell's own repair mechanisms to effect the type of mutation required. A simple demonstration of what happens is shown in the slide. Most of the time, and at all phases of the cell cycle, the cell has non-homologous end joining available. This means the cell recognizes that a cut has occurred and simply joins the two cut ends together. Most of the time, this results in error-free repair and no mutations. In a small number of cases, the result is error-prone repair, leading to insertions or deletions at the site, which in turn means a heritable permanent change in the sequence, and therefore, hopefully, a frame shift. This is then a simple knockout. In the S and G2 phases of the cell cycle, however, the sister chromatid becomes available as a template for repair, and the cell can use homology-dependent repair to fix the cut. If enough of a plasmid or simple oligonucleotide bearing resemblance to the sister chromatid by the addition of homology arms is provided, the cell can be made to insert the sequence of choice into the cut site. This is then a knock-in or gene insertion. But breaks induce DNA repair and increase mutagenesis frequencies by over 1,000-fold. In terms of what is required for genome editing using CRISPR, the CRISPR system consists of two main parts: the Cas9 protein, shown here in green, the guide RNA, which you can see here in purple, Now, the ribonucleoprotein, which is what the complex of the two is commonly referred to, glides along the DNA looking for a sequence complementary to the first 20 or so bases of the guide RNA. It stops at every protospacer adjacent motif or PAM site and checks the 20 bases adjacent to it. And when it finds the correct complementary sequence, the guide RNA binds and unwinds the DNA, allowing the Cas9 protein to cut exactly 3 bases in from the PAM. As I said earlier, we have a long history in this area and have amassed the most comprehensive suite of CRISPR products on the market. We have all-in-one plasmids, separate plasmids for Cas9 and guide RNAs, lentiviral plasmids you can package yourself, or lentiparticles ready to go, as well as whole genome arrayed and pooled libraries. Panels and libraries are available for knockout, inhibition, and activation. This is not an exhaustive list, of course, and we have other products and services that are not included in this slide, but these are the most relevant to today's topic. However, what we will be focusing on today is on the far right, highlighted in purple, which is RNP, and more specifically, the synthetically manufactured RNA component of the RNP. When we talk about sgRNA, what we're referring to is both the two-part system, which has the two RNAs involved in CRISPR in CRISPR editing as two separate, easily orderable products that then need to be annealed in complex with the Cas9 protein. So that's the cRNA and the transactivating segment of the tracer RNA. And the one-part system, which is a one-part single guide RNA system, which has the two RNAs joined together using a stem loop structure that does not affect the ability to cut. In fact, as you hear the talk, you will find that in many cases, the single guide RNA does outperform the two-part system. There is, however, a place for both options, and we'll continue to offer both to our customers. I'll now hand over to Peter so he can talk you through his slides. Thank you very much to Peter for collaborating with us on this talk, and of course, with the work we've done in the past. Over.
Thank you, Gurpreet. Hi, I'm Peter Romanienko. I run the Genome Editing Service at Rutgers University, which is a state university in New Jersey with 2 medical schools, a dental school, a cancer center, and an extremely diverse array of research areas. I'll go through a few slides that outline our workflow, show some data we have gotten using synthetic sgRNA. In my summary slide, I'll touch on a few areas for which I won't show data, but I will discuss our general impression on how synthetic sgRNA seems, in part, to have improved our results. Currently, we use CRISPR-Cas9 to make mouse models, and we developed a workflow that involves testing CRISPR guide RNAs in a mouse cell line, which in turn advises us on how to design the donor oligo or targeting construct. And then finally, we microinject or electroporate the CRISPR reagents and donor template into zygotes. The methods, resources, and reagents we use are just how our approach has evolved over time. It doesn't mean that this is the best way. It works for us, and we are open to improving our workflow. So this slide lists the steps we take. First, we design our guide RNAs using the CRISPR tool, web tool, looking for guide RNAs with minimal off-target and reasonably good predicted activity that would cut in the vicinity of where we want to modify or knock into. We transfect the sgRNA into Cas9 expressing NIH3T3 cells and check activity by T7 assay. This gives us an idea of in vivo function and lets us know the synthesized reagent is biologically active and not degraded after we receive and resuspend them. So based on the T7 results, or proximity to where the best guide RNA cuts, we design our donor oligo or a targeting plasmid. Once these are ready, we electroporate if we use CRISPR RNPs or oligo donors, or microinject if we use RNPs and plasmid donors into mouse zygotes. Once founders are born, we genotype them, and when appropriate, analyze the alleles by NGS to determine how many alleles there are, how mosaic they may be. So this next slide simply shows the cell-based sgRNA validation we do. Initially, when we used the 2-part CRISPR RNA system, we would transfect non-complex CR and tracer RNA, mixed in optimum, then with lipofectamine, and place them on cells. We monitor transfection efficiency using either a GFP or an mCherry messenger RNA. So we essentially see 100% transfection the next day using a fluorescent microscope, and then we can harvest the DNA within 24 hours to do the T7 assay. I haven't tried to harvest earlier. So this is a figure I had taken from Adgene. I think everyone's familiar how T7 assay works. I'll just go through it to cover all the bases. So the red X's represent mutations, not necessarily the same mutations across the whole population of transfected cells. Some cells may have one allele or both mutated, or all of them if they're aneuploid. Some cells may have just accurately rejoined the cut ends or may not have been transfected at all, and therefore there was no cutting. The genomic DNA is amplified from the population, and the products are denatured and rehybridized to purposely create heteroduplex DNA. This allows different strands to anneal together where different mutations are now across from each other, and then they create targets for T7 endonuclease 1 digestion. If two wild-type strands reanneal, T7 would not cut them. Also, if two strands with identical mutations reanneal, T7 won't digest them. Heteroduplexes are cleaved, and when run on a gel, you see the cleaved fragments. The ratio of cleaved to uncut fragments can give you an estimation of a guide RNA efficiency. So this next slide shows the T7 assay done with the first set of CRISPR guide RNAs we received from Millipore Sigma as sgRNAs. We typically design and test 3 guide RNAs for each target, unless we only find 2 good potential guide RNA sites, as in the case on the far right side for the gene PIT1. Rosa 26, to the left of that, is our go-to positive control. Is very consistent. These 2 lanes labeled CRC42 and tRNA do not give T7 cleavage products. Wild-type sample can give different levels of background, but hopefully the T7 bands are stronger than the non-specific cleavage. The lanes labeled under Rosa 26, 1 day, 2 day, and 3 day, which is not labeled, all seem to be equally efficient. So depending on, it doesn't matter when we harvest after the transfection. So the guide RNAs for the 4 targets represented on this gel all worked well, and we were able to pick the one for each project that we hoped would give the best positioning of the donor oligo to put in a mutation, allows to modify the PAM, and hopefully even introduce a restriction site for easier screening and have a high likelihood of success. So riding along on that high, I ordered the next set of sgRNAs and look forward to the results. So this slide shows, these results did not really give me a warm, fuzzy feeling. The Rosa 26 control worked, as you can see in the middle of the gel, and it always does, but the results for the others ran the gamut from bad to terrible. I had them resynthesized, and the repeat results pretty much mirrored these, so it wasn't an issue of quality, just perhaps limitations of the assay or the target locus was perhaps less accessible for the CRISPR-Cas9 complex. So in this case, we didn't have complete choice over which one we could use. These all were for inserting LOX-P sites, so we did have flexibility because it was not like we wanted to target a specific amino acid or were constrained by distance from the cut site. So we were unaccustomed to getting results like this. We kind of did not have great confidence in success of the final project. Ultimately, the projects worked, and in vivo cutting efficiency in the zygotes, as we surmised by founder genotyping, seemed to reflect this validation we do. We got the end result we wanted, but not at high percentage. But we found as long as we do see indels in the cell line validation, the project had a successful conclusion. We had ordered a few hundred more sgRNAs over the last few years and used them successfully in projects. All the projects worked, even with varying degrees of apparent sgRNA efficiency seen in our T7 validation, whether they were microinjected or electroporated. So one day Gurpreet asked if I would be interested in trying full-length synthetic sgRNA, and the results on this slide always stuck in my head. So I said, sure, I have a project to insert LOX-P sites. I'll try. cRNA and sgRNA, synthetic sgRNA, side by side, first in validation in cells, and then eventually when we electroporate the zygotes. So this slide shows a comparison of cRNA and synthetic sgRNA in cells. If you look to the right side, we see rows of 26 control working as lovely as ever, and the CR and synthetic sgRNA worked with equal efficiency. When I looked at the two target sites, the 5' LOXP target and the 3' target site, I was pretty much blown away. I repeated the transfection, the results were similar the second time around. I never get really sold very quickly on using a new technology or a new reagent with regards to CRISPR gene editing. But the synthetic sgRNA really sold me. Not because it would always be better, but because the rows of cRNA worked as well as the synthetic sgRNA. But I felt at least there may be more consistency project to project, seeing that in this case, all 6 sgRNAs clearly outperformed the cRNAs. We did not actually compare making the mouse model with cRNA versus synthetic sgRNA based on these results, because I found it hard to justify comparing the two reagents in a fairly critical project for this investigator. In one instance, though, we could have a reasonable comparison between CR and synthetic sgRNA, and that is in the targeting we did to the rows of 26 locus. Using CR rows of 26, labeled CRC42, we would get 10 to 15, maybe 20% knock-in at the rows of 26 locus, putting in different cDNAs. And this is using a very common targeting vector available from Adgene that have the traditional 5' and long 3' homology arms. With synthetic sgRNA, we saw targeting efficiency climb to 50% and higher. It is not a perfectly controlled comparison using rows of 26 CR alone. No targeting vector, we would see 70 to 80% frequency of indels at the locus. The synthetic sgRNA would give us a higher percentage indels and very often biallelic mutations. So we saw higher levels of targeting, but we would also see more biallelic mutations if we just wanted to look at indels at the rows of locus. So in the next few slides, I'll show some examples of biallelic editing, which we see, we seem to feel we see much more often with synthetic sgRNA, with the caveat that these are not side-by-side comparisons with CR and synthetic sgRNA. So this is a gel T7 analysis of founders for one project. The founders marked with an asterisk were chosen for NGS analysis, not necessarily hoping that they were heterozygous, meaning an indel across from wild type. Although based on the T7 assay, we knew at least the two alleles were different. So this is a readout of the NGS we get from the NGS service we use. The number of reads are usually in the tens of thousands. So we get the depth to clearly say we have all the alleles identified in this one founder, unless the deletions are large and excluded from the results. The top two alignments are two alleles in one founder. They have the highest number of reads. The rest are usually the same alleles, but with PCR artifacts and mismatches put in by Taq polymerase. So this slide shows the summary we send to investigators. We sort through all that data. The founder numbers on the left are listed on the left, and the mutations seen in their frequency are indicated. In this instance, the founders that looked to be possibly heterozygous by T7 assay had no wild-type alleles. I mean, this is not a surprise. This gene is not essential, but we would have expected some wild-type alleles based on our previous experience with cRNA, although there is no reason to absolutely expect that when we see the T7 results. So this next slide is just another example of biallelic editing where we did not see any wild-type alleles in the founders as analyzed by NGS. We did not bias our selection for founders that had biallelic editing. And again, this gene is not essential, although knockouts do have a distinct metabolic phenotype. At this point, we assume we get biallelic editing when we use a good sgRNA based on our T7 validation in cells. We would actually prefer to have indels across from a wild-type allele. Maybe synthetic sgRNAs are too good, if anything, And I'll discuss some ways later to address this, of which I'm sure many of you are aware or use yourselves. So this next slide shows a similar result, again, about biallelic editing, but this project is for an amino acid change. And in founders we chose, again, with no bias looking or wanting to get biallelic events, there were no wild-type alleles. Here we don't know if the amino acid change could do it in a founder. Perhaps it could present as a dominant allele or affect the founder's ability to reach sexual maturity and not be able to transmit the allele. We do like the efficiency in editing, but it can't, we can have too much of a good thing sometimes. So on my last slide, which summarizes some of our ideas, I'll go through our observations using synthetic sgRNA versus cRNA. So number one, I think there's an increase in cutting efficiency with sgRNA over the two-part system. From the T7 assay, directly comparing CR to synthetic sgRNA, on the one slide I showed, it was clear that synthetic sgRNA worked better in the assay, but the rows of control looked similar regardless of the source. Since that one time, and having converted to using only synthetic sgRNA, almost every target gave us 3 functioning guide RNAs. Sometimes only 2 work. This could be attributed to degradation after suspension, or the sequence targeted in NIH3T3 cells is not the same as the B6 reference sequence that I used to design the guide RNAs. And there have been a few instances over the years where the sequence we want to target does not even amplify from the NIH3T3 line. So we resort to in vitro digestion to at least know that this synthetic sgRNA is biologically active. So number 2, the increase in cutting efficiency may lead to more biallelic events. This is not an absolute belief, but we almost always find founders have biallelic events. In addition to this, we have seen very good targeting efficiency of plasmid donors to every target we have visited. We use reasonably short, although not extremely short, homology arms on our plasmids. We generally go with around 700-800 base pairs on each arm. And these are knock-ins of CRE, RTTA, or fluorescent proteins. So we get very good targeting, and it seems much more using synthetic sgRNA. So biallelic events can be good. Knock-ins to rows of 26, for instance, unless a loss of gene function is lethal. We do see biallelic knock-ins, like in the rows of 26 examples I referenced on a previous slide, and in plasmid knock-in to other loci. We've made a number of COVID-19 related models recently in the past few months, all with plasmid-based targeting vectors. In each case, the targeting went very well, and we often saw biallelic targeting events. Number 4, delivery can be adjusted to effectively reduce cutting efficiency. For example, prepare lower concentrations of RNP in microjection or electroporation mixes. So we have had to do this in a few instances. It is sometimes a priori not possible to know if a certain concentration will give you biallelic null alleles or no founders born due to knockout lethality. But a PubMed search and checking the JAX-MGI website can perhaps inform us about expected phenotype, looking at previous models generated or expression patterns. If we don't expect lethality, we like to go with about 50 nanograms per microliter protein and a 6-fold molar excess of synthetic sgRNA in our RNP preparation. We will go as high as 100 nanograms per microliter protein when putting in LOX-P sites. This may be too high. We've seen biallelic editing at lower concentrations. The lowest we have gone is 2 nanograms per microliter of Cas9 protein and have had enough editing to accomplish the project goal. This is just a brief overview of our experiences using synthetic sgRNA. I think it has been beneficial to our work, although we have not done a definitive parallel comparison. I cannot say this is the only way or indeed the best way to do things. It works for us, and we are always looking for ways to improve our efficiency and productivity. I would be happy to answer any questions later in the webinar or offline afterwards at any time. So thanks for listening, and Gurpreet, back to you.
Thank you for that excellent presentation, Peter. And I'm sure you'll have prompted some consideration and ideas with those results. and some follow-up questions for later. Just as a follow-up to that, luckily good timing from my point of view, we actually got a text from one of our customers in Australia literally last night, basically echoing exactly what Peter said there. So I won't mention the gene, but he basically said your gene guides were highly active in vivo, got 68% knockout mice, including 55% homozygous. So single guide do work extremely well in terms of animal models, just as Peter said. And then following on from that, the next set of experiments that I'm going to show you were designed to test how our synthetic guide RNAs compared with those of our competitors in a mouse model setting. To do this, 8 different gene targets were selected and were assessed for gene editing efficiency using our guides, as well as guides from 2 of our competitors. In this set of experiments, the guides were delivered by either in embryo microinjection or embryo electroporation, and the editing was assessed again by NGS. In the first set of data shown here, what you can see is that the percentage of CRISPR-modified alleles, where 15 embryos were microinjected with guides targeting the SARM1 gene. In this example, the guides from Merck showed an average editing efficiency of about 50%, whereas when the competitors' guides were used, that average was only 15%, even if the results were more consistent. At this target site, Merck guides clearly outperformed the competitors. On the graph on the right, we're showing the average data across all 8 different target sites, and this includes SARM1, and collectively over 250 embryos to compare the editing efficiency of our guides versus the. What the aggregated data shows is that on average, our guides perform equally as well as that of our competitors, or even slightly better in some cases. We also performed the same set of experiments in the context of embryo electroporation. As we know, many of our customers are using this method of delivery for CRISPR reagents. Again, using SARM1 as an example, our guides showed an average yet highly consistent editing efficiency of 93%, as compared with our competitors who only showed 42%. Shown to the right is again the average across all 8 different target sites in a collective total of almost 250 embryos, where either our guides or competitors' guides were used. What we can see here is that again, and even more so in this case, our guides performed equally as well, if not slightly better. Overall, our single guide editing efficiency in embryo electroporation and microinjection studies performed equally as well and even better in some cases than ours. So moving on then to single guide RNA and cell-based assays. Before I introduce this data, let me give some background to this particular experiment. So we work with Chandra Miller at St. Jude's Hospital on a large collaboration that involved 6 different targets in K562 and U2OS cells. She did several comparisons. Number 1, comparison between 2-part unmodified and modified, 1-part unmodified and modified RNA. 2, a comparison of a couple of different modification patterns, which ultimately helped us settle on what we offer today. And 3, comparison of our RNA versus a competitor. The conclusion over the whole set of experiments was that in her hands, modified single guide RNA worked the best, and that ours worked extremely well when compared to others. The data you see on this slide is the last data set that we worked on with her, and the purpose of this experiment was to see whether titrating up the amount of cRNA trace RNA would improve the cutting efficiency up to the levels of modified single guide RNA. One note on the experimental setup: their previous experiments had been conducted at about 1:8 ratio Cas9 to guide RNA, so they included one data point at that concentration as a control. But the titration experiment is done at a 1:2 ratio, as they had done multiple experiments by then to show that 1:2 and 1:3 ratio is more efficient for most guide RNAs in cell lines that they tested. The take-home message really, as you can see from the data itself, is even at the highest concentrations of cRNA and trace RNA, they were not able to get the same activity as they see with the single guide RNA modified at the lowest concentration tested. And they probably could go lower to get that same activity. One additional point from me You can see that the cRNA works very well, right? So you're talking about 40% up to about 70%, 80% cutting efficiency. It's been around a good while and is at a slightly lower price point and is an established and published product that has been demonstrated to work across the board in both cell lines and animal models. Hence our decision to continue selling it. However, if you need the extra consistency and higher cutting efficiency, then single guide RNA is definitely the way to go. This experiment was done by Andrew Bassett at the Wellcome Trust Sanger Institute. We really wanted to have an evaluation done in iPS cells, and Andrew has a great iPS model which has one copy of BFP integrated at the ROSA 26 locus, which when altered by basically just 2 bases, switches the BFP to GFP. Whereas the previous set of experiments were all done for knockout, this experiment is a simple knock-in with a single-stranded oligo donor to alter those 2 bases. Andrew used a hi-fi Cas9 that they make themselves in-house. The cRNA and trace RNA is from a competitor and is the modified single guide RNA on the far right. And of course, the one in the middle is the modified single guide RNA here from Merck. On the far left, you can see the negative controls with 0 GFP positive cells and 94.7% BFP positive cells. From the results, you can see that they were able to get 38.5% GFP positive cells with the 2-part system, but an increase to 45.6 or 46.1 when they used modified single guide RNA, showing nicely the utility in iPS cells of single guide RNA and the improvement of knock-in of using single guide versus 2-part RNA.